Ms. Patterson here. Today we're going to go over Meiosis, AP Biology, Unit 5. So when we talk about meiosis, we need to talk about the chromosomes. And chromosomes are genes, and you get one from mom and you get one from dad. These are going to be segments of DNA that code for traits that are passed from one generation to another. So your hair color, your eye color, your skin tone, your height is all determined based on genes of your ancestors from your mom's side or your dad's side. Now we have what are called gametes, and gametes are going to be your sex cells. So that's what's going to carry these genes, and that's going to be egg and sperm. Animals have egg and sperm, and so do plants. And this is going to create a varied type of population due to the mixing and the passing down of these genes. On each chromosome, you have what's called a locus. So this is a chromosome, and these yellow lines are what we call a locus. And these locus are kind of similar on every chromosome. So let's pretend this is chromosome 1. In chromosome 1, we would say everybody's chromosome 1 in this area of your chromosome would represent eye color. And on everybody's chromosome 1, this could represent height. And chromosome 1 in everybody, this could be the development of your small intestines. This one could be pigmentation. So everybody will have these chromosomes in very specific sections on your DNA, and they're numbered 1 through 22, and then you have the 6 chromosomes. We'll talk about those in a second. But chromosome 1 we're just going to pretend that that's what this chromosome 1 would have, and everybody's chromosome 1 would have these traits in similar areas. There are different types of reproduction. You have asexual and sexual. Asexual is when a parent basically makes a clone of itself. So here I have a C polyp, and it's going to create a carbon copy. It's just basically going to start doing mitosis, where it's just making carbon copies of itself. And then the plant will detach, and then this will float off and go to another part of the C and embed. But this is going to be an exact carbon copy of the parent. The only way I can have any type of variation is if there's some type of mutation in the developing process of this type of plant. Now, from this point on, if this piece here became mutated, all of this offspring would be mutated because that code has now been embedded into the genes or the traits of this specific plant. Sexual reproduction involves 2 individuals, and they're going to contribute genetic material to offspring. This is very good for expanding a population and creating a healthier population by changing up the gene pool. You're creating more variation. So when you have sexual reproduction, you have an egg and you have a sperm, and this is going to have your genetic information, your DNA. These will fuse together, and this will create what's called a zygote. And the zygote will then develop into a baby, into a frog, into an elephant. Whatever this gets coded for is what it's going to start to create. So there's 2 different types of cells we need to know. One is called somatic, and these are body cells, and these are going to be like skin, eye, hair, intestinal lining cells, cells inside your mouth. These are all body cells. And these are diploid, which means I have both sets of chromosomes. I have a set from my mom, and I have a set from my dad. So these 2 sets in humans are going to total 46. I get 23 from mom, 23 from dad, and that gives me 46 chromosomes. This is now part of my genome, or part of my genetic makeup. Gametic cells are your sex cells, and that's either going to be egg or sperm. And these are haploid. They're going to be half this number. And the reason I need to make sure these sex cells, these gamete cells, are half is because if I have an egg or a sperm that has 46 chromosomes, this egg has 46, this sperm has 46, I'm going to have something that's going to have 92 chromosomes. Humans don't have 92. We have 46. So for me to get 46, I have to have half that number for the egg and half that number for the sperm. So this is haploid, which means half. So I get 23, or half of whatever that organism is. If I have a insect that only has 20 chromosomes, it's a diploid of 20, then each egg and each sperm will have 10. So it's kind of thinking of that way. I've got to have half of that number for me to make my whole chromosome diploid group. Homologous chromosomes, homo means the same, but these are going to be chromosomes that are going to carry the same genetic information. So here again, let's go back to chromosome 1. This is chromosome 1 for your dad. This is chromosome 1 for your mom. So this top line could be eye color, this could be height, this could be small intestines formation, this could be pigmentation. So in your mom and your dad, you have chromosomes in similar areas. Now your dad will go in and he will make sister chromatids. When he replicates, he's going to have one half of this chromosome. It's going to replicate and create what are called sister chromatids. And your mom's chromosomes are going to do the same thing. And then later on, they're going to kind of fuse, they're going to come together and they're going to start swapping information back and forth. And this is what gives variation in siblings. So if you have the same mom and dad, not all of your brothers and sisters look the same. If you have the same mom but different dads, you know, your offspring siblings are going to look a lot different probably. So this is kind of creates variation in it. The only time you don't see that is with identical twins, and that's a whole different thing. This is what's called a karyotype. This is an image that shows your complete chromosome pairs. So again, we have 46 chromosomes because we have 23 sets of chromosomes. So we have 22 which are called homologous pairs, or they're your autosomal pairs. And then I have my sex set right here. If it's XX, you're female. If it's XY, you're male. So this is a genetic marker for gender. This right here is going to be similar in most everybody. These are all going to have similar, like again, if this was chromosome 1, this would be eye color, height, intestinal lining makeup, pigmentation. So this type of information would be similar on everybody's chromosome 1. One would be from your mom, one would be from your dad. Chromosome 2 would have very specific traits, 3, so on and so forth. So with your gender, your mom is always going to donate an X. That's why everybody has that X chromosome. Now males have an X or a Y. So when they are donating one of their sex gametes, it depends. If they donate the X, you're going to be female. If they donate the Y, you're going to be male. So it's the male that determines the gender of offspring. Here's another karyotype. You can see the locus points here that show similar characteristics for gene traits. And you can see all of these autosomal chromosomes, and then here are the gender or the sex chromosomes. When something becomes fertilized is when one egg and one sperm fuse to form a zygote. So this genetic information from the egg, this genetic information from the sperm come together to make a zygote. These are haploid. When they fuse, I now become diploid. And then this will start to create the coding to build the organism, whatever it is that it has to build. Now with meiosis, the best way for you guys to remember the difference between meiosis and mitosis is this first right here, me. Okay, I have to think about how was I formed. I was formed with parents, and so I'm going to have an egg and a sperm fused together and formed me. Mitosis is just repair, replace, and to grow. So again, if I cut my hand, okay, I'm going to start to create skin cells to replace it. I'm not creating stomach cells. I'm not creating eye cells. Like I'm creating more skin cells. I'm not creating anything new. Where with meiosis, I'm creating something new. I'm creating something varied. So if you remember that, that helps you kind of remember the difference between the two. So with meiosis, I have a combination of gametes that are randomly going to distribute genes, and it's going to reduce the number of chromosomes. And we'll talk more about that here in a second. Because remember, I don't want 92 chromosomes. I need 46. So I have to be able to split down and kind of break down my chromosomes to where I end up with 23 and not 46 for humans. In the end, all offspring are going to have the same number of chromosomes as the parents. Now, there are some genetic problems that can happen, Down syndrome's one, where they actually have an extra chromosome. But those are genetic issues that can kind of arise during meiosis. So let's look and compare meiosis and mitosis. So mitosis is going to replicate DNA just like meiosis. But with mitosis, I'm going to have identical cells. I have a mother cell and a daughter cell, and they're going to be identical. Whereas in meiosis, I'm going to have varied and different cells as the outcome. With mitosis, I have one stage of cell division. With meiosis, I have two. With mitosis, I'm just only going to have two daughter cells, and each are going to be diploid. So if these were human cells, I would have 46 in each cell. Here, I get 4 daughter cells, but each one is going to be haploid. So if this was human, this would be, each cell would have 23 chromosomes. And again, this is just going to be egg, it's just going to be sperm. So when we look at interphase, this is going to be the part of the process where I'm going to have replication of DNA starting, I'm copying my chromosomes, I'm checking to make sure everything's ready to go. And when I have that, my DNA is going to be very spread out. It's going to be very relaxed. It's going in, it's doing its job, it's copying, it's making proteins. But when I'm getting ready to multiply, I'm going to start to condense, and I'm going to start to pull all of these strings together. I'm getting ready to move. And if I'm getting ready to move, I need to pack everything up. And so all these chromosomes are going to start to get real thick, and it looks like spaghetti or kind of really wormy inside that as all this spread out chromosomes come together. I'm going to have 2 identical sister chromatids. So this, let's say, are the dad chromosomes. These are the mom chromosomes. This is going to be chromosome 1, and I'm going to make a replica of it. So dad chromosome 1, copy of chromosome 1. These are sister chromatids. Mom chromosome 1, copy of mom chromosome 1. These are sister chromatids. Okay, these would be homologous chromosomes because these will all be chromosome 1s, but I'm going to have on the mom, I'm going to have the dad. So here I have my DNA, it's replicated, I've made doubles. Now I'm going to mix up, so remember there's 2 stages in meiosis. This is going to mix up and it's going to start to blend some of these chromosomes. Here I have them mixed up, but now I need to spread them back out to 23. So for humans, this one would have 23, 23, 23, 23. So I have to split them up from 46 and break them down into that. Because again, if this was 46 and I fused with another gamete that was 46, I would have 92, and I can't have that. So I need to make sure these are always half of what my haploid or my diploid cell needs. So meiosis 1, this is going to be the first division with a diploid cell. So these are going to, homologous chromosomes are going to come together, they're going to mix, and then I'm going to have these cells that are going to have 46 here. Now they'll tell you that these are haploid because they're splitting, but these still have 46 chromosomes. So prophase 1, the chromosomes condense. The sister chromosomes are attached at the centromere. So remember, centromere is here in the middle. I'm going to have synapsis, which is going to create what are called tetrads. And what happens, this is mom chromosome 1, we'll say this is dad chromosome 1. They are going to line up, and they basically like Velcro. They're going to attach to each other. And this is super important when we talk about the mixing of these traits here in just a second. They're going to attach and stick to each other. So this is where we're going to have crossing over. Since they're stuck together like Velcro, the pieces are just going to basically flip, and they're going to just trade places. So on these different locus points, I could have an area where it's eye color, and then they're lined up right next to each other, and then the mom side and the dad side will just switch. So now the mom's over here on the dad's side, the dad's over here on the mom's side. I just randomly swapped out some of these different pieces. And again, this is just a random switching of material, which is called crossing over. So these homologs, which are these homologous chromosomes, are going to start switching these places. And these occur at what's called the chiasmata, which is right here, kind of where the parts kind of attach and pop around. So now this pink was over here on this side and this was over here, and they just flipped and just changed sides. So now I have a little random just mixing of material for both ones. So now my homologous chromosomes are a lot different. I have metaphase 1. This is going to be again another random displacement of chromosomes. They are going to line up in that metaphase plate. So remember, it's the center. They're all going to line up similar to like mitosis. And it's just going to be random. They're just going to randomly just line up. There's no rhyme or reason. It'd be like going with a group of people and saying, I need everybody to make a line. People are just going to get in line. And then you told everybody, all right, spread out. Everybody get in line. The line's going to be different every time. And it works the same way with independent assortment. These are just going to randomly line up in different ways. So I could have a line that looks like that. Another one could look like that. Another one. And another one. I can have many different variations of this. So now when they get ready to split, this side will go one way, this side will go the other way. And if you look, I'm going to have a different variation every single time. So each cell is going to be different. So here I have random assortment. They're lined up on this metaphase plate. I'm going to have one group go one way, one group is randomly going to go the other direction. In anaphase 1, they're going to start going to opposite ends. I'm going to have these spindle fibers are going to start pulling to one end of the cell. The sister chromatids are going to stay connected for right now as they go away, but they're going to start to separate. Now I have telophase 1 and cytokinesis. These homologous chromosomes are going to move to the opposite ends of the cell. Each chromosome is still made up of these two sister chromatids for right now, so they're still attached at this point. So now I'm going to have two separate cells, okay? Since the chromosomes have been cut in half, they call it haploid, but my number is still going to be 46 for right now. All right, so this is meiosis 1. I do prophase, metaphase, anaphase, and telophase. Sometimes you'll hear it's called telophase. But this part of meiosis 1 is all about mixing. I'm mixing in prophase. I'm mixing in metaphase. I'm pulling apart in anaphase. And so all of this is, the whole purpose of meiosis 1 is to mix and to vary genetic offspring's traits. So now meiosis 2 is going from this large number of chromosomes to go into a true half chromosome. I need a true haploid cell. So now I'm going to have my 46, 46. I'm going to line up again on a metaphase plate. And again, I'm randomly going to get pulled to either side. Anaphase is going to go through, and then I'm going to have telophase is going to come in and create my cells. So now I'm going to have 4 cells, and they're both going to be individually different due to all the stuff that happened in metaphase 1. All right, so prophase 2, my spindle fibers are going to form again. Sister chromatids are going to move towards this metaphase plate. But you can see now, this is going to split. So this one is going to go one way, this one's going to go another way. I have completely different types of chromosome setup here. My configuration is going to be completely different for all of these. So the sister chromatids are going to line up on this metaphase plate. They're no longer identical because of all the variation that happened in meiosis 1. The spindle fibers are going to come back, they're going to attach to these kinetochores, and they're getting ready to pull apart. Anaphase 2, they're going to disappear. The chromosomes are now going to pull to opposite ends of the cell in both of these cells. Telophase 2, I'm going to rebuild my nuclear membrane. I'm going to have completely two different cells. And then cytokinesis is going to make sure all of my fluid, all of my material is equal on both sides. When I'm done with telophase 2 and cytokinesis, I'm going to have 4 individual, unique daughter cells. Each single one is going to be different. This is why it's so hard to find somebody that looks just like you. There's always going to be some type of variation in your DNA because of all of this mixing and randomness. So meiosis provides that genetic diversity. It's going to produce variation in offspring, which is good. It shows a healthy population. Crossing over is going to happen in prophase 1. Independent assortment is going to happen in metaphase 1. And in metaphase 1, this is amazing, 8.4 million possible combinations with your chromosomes in metaphase 1. Just lining up on that plate. That's how many different possible chromosomes there could be. Now, with random fertilization, because remember, this could be individual egg, you could have individual sperm. So I have one egg here, and this is one of 8.4 million possible combinations that happens to fuse with a sperm, which is one of 8.4 million possible combinations. And that's why you can have 70 trillion possible combinations. And that's not really even considering crossing over, doing all of this other stuff. This is why you don't have a lot of people that are identical. Now, you'll see people like the doppelgangers that look similar, but genetically, you're going to be able to see that they're different. All right, that's meiosis, AP Biology. I hope that was helpful. If so, let me know, and I hope you like and subscribe. I'll talk to you guys later.